Hey guys, so we're going to finish off our notes in the end of chapter, 16 here. Before you even really get into this video, I just want to give you guys a heads up. So in Google Classroom, where you found this video, there's also something labeled Genetic Technologies Videos. In there, I've listed about 6 different YouTube videos that kind of explain what we've been talking about and to give you a little extra information. If you could pause this video and go back to that, I would like you to watch the videos on gene regulation. I believe that's number 6. And it talks a little bit more about gene inhibition, operons and operators, and gene activation. I've also listed in there a gel electrophoresis video, and I have also put in a video on GMOs, so genetically modified organisms, and genetic engineering, which is CRISPR. So there's quite a few of them. The only one I really kind of want you to watch is the gene regulation at this moment, just because it might help you understand the process that we talked about earlier on. All of those videos are really good, informative pieces that I would watch at some point and then come back to this. You don't have to watch them all, but they might give you a little bit more information. So what I'm going to do now in this video is I'm going to finish off our notes with you. So if you could go back to chapter 18, that says chapter 18 technologies, and it says cloning. We're going to start with cloning. Once I'm done talking about cloning, I would like you to go watch the cloning video. It's about 3 minutes long. And then I'm going to finish off with PCR and gene therapy, and you'll have to just watch the PCR video as well. So before we even talk about cloning, we have to talk about the fact that asexual reproduction is the natural form of cloning. So asexual reproduction gave rise to cloning. So if you're looking here, it says, what is asexual reproduction? Well, essentially, asexual reproduction is the naturally producing identical copies of a parent cell via mitosis, essentially. So we're producing genetically identical organisms, okay? One parent, 2 genetically identical children. Binary fission of bacteria, budding of hydra, the propagation of plant cuttings, which you'll see in the video, vegetative spores, anything like that is considered asexual reproduction. So because of asexual reproduction, they started to look at it and say, hey, can humans do this? So that's when the cloning of Dolly came about. In 1996, they successfully cloned the first sheep, and her name was Dolly. So this is how they did it. So if you could fill in your notes, it says cloning of Dolly. I would just like you to follow along, okay? So how did they do it? Well, first they took a haploid nucleus and they removed it from an egg cell of a surrogate sheep. Please underline, highlight the word enucleated egg. That shows up on the diploma often. Enucleated egg means no nucleus. They remove the nucleus. Okay, so we took out 23 of the chromosomes from the egg, and then we borrowed a diploid nucleus from an animal that we want to be cloned. So the diploid mammary cell from an animal to be cloned is then inserted into the enucleated egg. So we removed the haploid nucleus, then inserted a diploid nucleus. That's essentially what step 1 and 2 are saying. Take out the haploid, insert the diploid. They are from 2 different animals. Okay, so they're not the same sheep. It's sheep A and sheep B, and that's how they started. So sheep A donates the egg. We remove the nucleus. Sheep B gives us probably a skin cell of some sort to be put into the egg. Once we have reinserted the new nucleus in, the egg undergoes electric shock therapy, which essentially triggers a cell division. That cell division then allows for an embryo to start developing in vitro, so in a petri dish, essentially, okay? Once it is developed in that petri dish, we then know it's at the blastula stage. We take it and we implant it into a surrogate sheep, which is going to be sheep C, essentially, okay? So sheep C is going to then grow this little baby sheep, and eventually we're going to pop out a baby, and that baby is going to be identical to sheep B. So sheep A donated the haploid nucleus. We removed it. Sheep B gave us a full set of DNA. So because we have a full set of DNA, we now have an identical copy when sheep C gives birth. Okay? So that is how Dolly came about. So here is the picture. What you're going to see here is the enucleated egg, and here's the mammary cell. So this is sheep A, sheep B. Okay? We donate, okay? We take this inserted here, remove this, make a blastocyst, implant it into sheep C, and then there you go. You have Dolly, who's identical to this person up here. Okay? So that's how cloning Dolly came about. Now, here was the thing though. They cloned Dolly and Dolly lived for a while, but what ended up happening was Dolly died younger than what her parents did. So that's Dolly and her mom. Okay. So Dolly grew up, but she didn't live as long as her parents did, even though they had identical sets of as long as her mother did. And that's because they had identical sets of copies. And scientists were like, why? What's going on? So what they ended up doing is they ended up looking at something called telomeres. And I just talked about this with the Fragile X syndrome. Okay. Telomeres are little caps or repeating sequences of A's and T's on the ends of your chromosomes. And every time we go through mitosis, it actually shortens the telomere. Okay. Now, telomeres are considered to be linked to aging. So as we shorten our telomeres, we have less and less of time for our life, for our chromosomes and all that stuff. Now, here's why Dolly didn't live as long. Although Dolly was an identical copy of her mom and was born as a baby, her genetic information was a 6-year-old sheep because she was identical to mom. So the telomeres on the ends of the chromosomes were actually 6 years old. So Dolly, at a baby, genetically was 6 years old. So she didn't live as long because she genetically was already older. So in that section, you need to paraphrase what I'm saying here. So essentially, Dolly died young because her telomeres were old and she didn't get to live as long. Okay? So that is one of the downfalls to cloning, is we do not know how to stop the telomeres from shortening. Otherwise, if we did, then we could live forever, right? So what they did is they said, how can we apply cloning to everyday life? We applied it in the sense of Dolly. So what are other special abilities we have? So plants, they realized, have the special ability to reproduce themselves from any cutting. You cut off a piece of the plant, you dip it in some hormone growth stuff and you plant it, it's going to produce an identical copy of the plant. This is because plants have something called totipotency. Totipotency means that they are not differentiated. Please write that down. Totipotency means they have no specific job. There's no differentiation. Now that you've written that down, we can look at it and say, well, our skin cells are not totipotent. Our skin cells are our skin cells. Our liver cells are our liver cells. Can humans do this? And the answer is yes, but our cells are only totipotent in days 4 to 6 of pregnancy when we have a blastula. So that initial embryo stage, we call this stem cells. Stem cells are undifferentiated. They are totipotent. So to look at it, we have to realize that cloning and genetic engineering and stem cell research all come from this idea of understanding mitosis, but in the fact of humans is we don't have totipotent cells unless you're an embryo. So that's when the ethical side of everything arises. Okay? So stem cell research, which you don't have anything to write down with, you can put it off on the side, is kind of mixed in with this. So at this point, I want you to stop, pause this video, open up the YouTube video on cloning, and please watch that video to understand what I just paraphrased in your notes. So stem cell research. Again, you don't have this in your notes, but I'm just going to quickly talk about it so that you can kind of see where this idea of genetic engineering and cloning came from. So again, stem cells are capable of replicating, but they're not differentiated, meaning they can be any type of cell they want to be. They're kind of like you in kindergarten when you think you want to be a flamingo or a birthday cake or a dancer or a fireman or a police officer, or heck, maybe you wanted to be a sanitary engineer when you grew up. I don't know. That's what cells are like. They're not differentiated. They don't have a set role, okay? We call this pluripotent. We get stem cells 3 different ways in our bodies. The embryo, which is the blastula, the umbilical cord, which is why umbilical cord blood donations are very important. When you give birth, you can do that. You can save blood from the umbilical cord because it has cells in it that are pluripotent, which are not differentiated. And so if your child ever becomes sick, you can use those stem cells to potentially help cure cancer or give them cells and replace cells that are sick. So that's a big one. Or in adults, our bone marrow, that's why bone marrow transplants are a big thing, are actually pluripotent. Okay, so in your notes where it says the special ability of plants to reproduce from any part of a plant is called totipotency, underneath where it says, can humans do this? Say yes, we get stem cells from, and then write these 3 things down. So eventually, stem cell research, their ultimate goal is to repair damaged tissues, to help fix maybe brains that are not functioning because dopamine neurons in Parkinson patients don't work. Or maybe they want to get skin tissues for burn victims, regenerate blood cells. Stem cells have a huge array, big one, helping people walk again, refixing spinal cords, have a huge array of application, but unfortunately, the ethical side of it is, can we do it? Is it right? Okay? So there's that. So again, with all of this research going on, are there any sort of, what are the, I guess, the ethical sides or the controversial sides of stem cells? So at this point, I don't have a video on stem cells, but we're going to skip through because we have talked about genetic engineering. So I'm just going to graze through this. At this point, you might want to stop the video and go and watch the genetic engineering videos. But we're going to move forward in your notes past genetic engineering, past isolating DNA, past gel electrophoresis, down to the forensics part and polymerase chain reactions. So here we are, forensics. Often DNA is found at a crime scene, which we talked about. What we're going to talk about is, we talked about how gel electrophoresis can be applied. Now we're going to talk about how polymerase chain reactions can be applied. Polymerase chain reactions, or PCR, are sometimes used with gel electrophoresis to create images. Okay. We want to use gel PCR because what it allows us to do is it actually allows us to take a small piece of DNA, and rather than use up that small piece of DNA and not have it anymore, it allows for us to make multiple copies of this DNA so that we can test it in many different ways. All right. The guy that came up with this was Dr. Edward Southern, and he called this process Southern blotting. So that last blank you have is called Southern blotting. So a video on PCR is loaded on that Genetic Technologies. It doesn't have any talking. It's just a sound that walks you through the process. So if you could please watch it, that would be right now. I am going to talk about it quickly. So if you want to wait, you can, or you can watch it and then watch this part. So PCR is when we need lots of copies of a gene to do an analysis. So essentially, it is an automated process or a human-run process of replication. So PCR takes the DNA fragments and we then essentially copy it. And then it's used for forensic purposes. Again, like I said, it is 100% like replication. And if you look at it on the side, you see the double-stranded DNA pulled apart. We got 2 primers, and then we use DNA polymerase, which at specific temperatures, if you're noticing the side, there's specific temperatures to do this. Then you copy the DNA and you repeat and repeat and repeat until you have enough copies of the DNA to do as many tests as needed. So we have already talked about gel electrophoresis in forensics. So the last thing I want to quickly jump ahead, and there is no video on this at all in the other section, is just gene therapy. Okay, so gene therapy is essentially something that has arisen to help treat genetic diseases. It's very much so still in its clinical stages at this point. There's a lot of stuff that has to go into it and a lot of testing, and obviously we don't do a ton of testing on humans. So it's essentially taking gene replacements and taking a good gene and replacing the defective gene with that good gene. They're hoping to possibly cure things like sickle cell anemia, diabetes, cystic fibrosis, all of these things where we can remove a bad gene and fix it with a good gene. So that is, you can read a little bit, and there's a picture and a diagram about it right here. This is in your notes, so you can kind of see how that works. And other than that, guys, those are the technologies. So your notes are filled out. You can watch these videos, and please start studying for your test. The videos in this should take you most of the class. You can do work on your stuff as well for your project. The videos are on there forever, so if you don't get to them today, you can watch them over the weekend. So I hope you guys have a great weekend, and I'll see you on Wednesday in room 712.